Hello and welcome to another ADLC digital lesson. Today's lesson is on the structure of DNA. When you look at a model of DNA, the structure can be very confusing. No matter how you twist or turn it, it can seem like a jumbled mess. If we're going to be able to understand DNA structure, what we really need to do is simplify it. So what we'll do is ignore the complex molecular structure for a second and just examine the shape. The shape of DNA is described as a double helix, which is kind of a twisted ladder shape. But we can simplify even further by untwisting the ladder. So now we see that DNA isn't so complex after all. It's just two sidebars with rungs in between. But we can even break things down more. The individual parts of DNA are really just repeating angle-shaped units. They are called nucleotides, but of course, they are not made of metal ladder. Nucleotides are made of three molecular parts: a nitrogen base, a sugar known as deoxyribose, and a phosphate group. So if we stick nucleotides together, we can start to see the DNA structure take shape again. At this point, we should note that nucleotides come in four different flavors, so to speak. While the sugar and phosphate don't change, there are four different nitrogen bases, shown here with different colors. They are adenine, thymine, cytosine, and guanine, often abbreviated by the letters A, T, C, and G. Here we have only four linked together, but they can form chains of thousands. But where is our ladder shape? Well, a string of nucleotides mirrors the first, with the nucleotides bonding together in the middle. And there's our ladder shape. There is one important rule to this bonding, though. Because of the shape of the nitrogen bases, A can only bond with T, and C can only bond with G. Any other base pairing is impossible in DNA. So if you know the bases on one side of the strand, you automatically know the other side. But we have to remember that these nucleotides really don't look like blocks and circles. If we return to a more realistic view, we can still see the nucleotides and how they fit together to make a complete DNA molecule. And now that we have a good understanding of the nucleotides that are at the heart of the DNA molecule, perhaps we can now return to the beginning and realize what first looked like a jumbled mess of a model is really not that complicated at all.